So, good afternoon. You may see that we have been waiting for a couple of minutes. I want to welcome you to the President's Symposium, which is traditionally the highlight of the first day, next to the other highlights that we have already since we started this morning. And this is the awards ceremony, and we are very happy that the awardees and their laureates and the laudationes are already here. We may start a little late, as you see, because the YI session started late because we were a bit late with our welcome note. So this is why we just decided we start now. The YI people will just come over in a couple of minutes, so when it's a bit sort of, you see people coming in, it's just the YI group that will then join us. But we decided for some reasons, because some of our guests have to leave earlier, that we will just now start. It is my great pleasure to, I just need my presentation, by the way. Exactly. So we will start with our lecture series on the awards that we have. And this ceremony is really important because what we do have is a selection of young and advanced immunologists who are awarded here at the Presidential Symposium. So the award ceremony will start with the Otto Westphal Thesis Prize. And you see here already the winner, and I'm very happy that Tobias Bob from Mainz will give the laudatio and tell you why, among many others with excellent work, we have a Sebastian Basch winner for the first award. Tobi, please come on stage and give your laudatio, and we were curious to hear for the selection of this laureate.
Okay, thank you very much, Christine. So on behalf of our president, it's my great pleasure to hold the laudatio for the Otto Westphal Prize, because you probably all know that promotion of young scientists is at the heart of the mission of our society. And the whole society, I have to say, is particularly happy that we share this interest in promotion of young scientists, also with companies, because something like that is not possible without a sponsor. And I would especially name here Biotest and thank Biotest for also sponsoring this prize. But now coming back to the election of this year's Otto Westphal Prize, we had 13 excellent, I have to really say excellent applications. The colleagues of mine that are in this selection committee know that. We had a tough time to really select one. But at the end, it was a quite clear decision, and you already heard that we decided to give this award to Sebastian Basch I learned from Freiburg, but it was kind of a joint project between Freiburg and Leipzig, but probably even happier to do so, because as you probably know, Otto Westphal was one of the founders of the Max Planck Institute in Freiburg, so in a way it all comes together, and he was sending in his application on his very excellent work on how cytomegaloviruses shape the phenotype and function of macrophages, resulting in altered or modulated immune responses to this virus. And without any further ado, I would like to welcome Sebastian and also a representative, I think, of Biotest, Ms. Where are you? Yeah. To hand over the prize, please.
We are very happy. So we are very happy that the company Biotest is sponsoring this award, and Mrs. Rastovic is here as representative of Biotest because they are especially dedicated in supporting young scientists, and this is exactly what we want to do. So thank you very much for coming. We took the pictures in advance so that we won't lose time here on the way of our ceremony. So we have the pictures already. You will get now your award, and we would like to ask you for a big applause. So. So now we will hear in your own words what was the work this was awarded with the Otto Westphal Prize. So please go to, this is your audience now to tell us why we think that this was really a great work. Thank you.
Weiter. Okay, perfect. Okay, hello everyone, good afternoon. First of all, I would like to heartily thank the awarding committee of the German Society for Immunology for awarding my thesis for this year's Otto Westphal Prize. Today I would like to share some results of my thesis in which we show, wait, we can do it like this maybe, in which we show that cytomegalovirus subverts macrophage identity. So as you know, macrophages are part of the innate immune system, and they basically populate all organs within our body. And to become a macrophage, we need cell-specific transcription factors that steer progenitors to differentiate into macrophages. Then we also need tissue-specific transcription factors, tissue-specific transcription factors that enable these immune cells to adapt and specify or specialize into immune cells that are resident in each specific tissue, for example, the intestine, the lung, or the skin. Generally, macrophages are known to maintain or important for maintaining tissue integrity and to protect from invading pathogens via phagocytosis and subsequent cytokine secretion to recruit other immune cells. Alveolar macrophages, for example, they reside in the alveolar space in the lung, and they're dependent on the transcription factors PO1 and, most importantly, PPAR gamma. They can be identified by the combination of surface markers C11c, cyclic F, and the Fc gamma receptor 1, CD64. Alveolar macrophages are important for the defense against airborne pathogens such as Legionella pneumophila, where they secrete cytokines to initiate the recruitment of neutrophils and to ultimately control the infection. Cytomegalovirus, on the other hand, is an opportunistic pathogen. It's a herpes virus that infects most children worldwide within the first year of life through the respiratory tract. And as you may realize, this infection occurs at a time point where the immune system of a neonatal infant needs to or faces a multitude of potentially colonizing but also potentially invading microorganisms. So during my doctoral thesis, I asked the question, what are the consequences of a postnatal CMV infection for the innate immune system and especially for macrophages? And to address this question, we infected mice intranasally with a mouse CMV and used combined host pathogen fate mapping. So here we use the C11c specific Cre recombinase to cleave the stop codon preceding of a viral and also host reporter fluorescence. Doing so, we saw that control, but also non-infected bystanding macrophages, as expected, expressed tomato positive, driven by the host ROSA26 promoter. Infected alveolar macrophages, which are predominantly targeted during respiratory tract infection with CMV, are labeled with both GFP and tomato. So now we were able to specifically sort these cells, these infected alveolar macrophages, and to sequence them. And we saw that their transcriptomic profile highly differed from those of bystanding and control alveolar macrophages, including the aforementioned transcription factors, and also on protein level, the surface marker C11c, cyclic F, CD64 were significantly downregulated, suggesting a loss of identity of alveolar macrophages. And indeed, again, using combined fate mapping, but this time of sorted alveolar macrophages, or of transfer, after transfer of sorted alveolar macrophages into CSF2 receptor knockout mice, which are devoid of their own alveolar macrophages, we saw that these cells basically left their niche. So they left the alveolar space. You can see here, tomato single positive, non-infected bystanding alveolar macrophages, a macrophage, and here is a CMV-infected alveolar macrophage that migrated into the interstitium, and there it spread the virus locally. And that was surprising because alveolar macrophages are considered to be resident and stationary once they found their tissue of choice, if you want. And we were even more intrigued when we saw that these GFP tomato double positive infected alveolar macrophages migrated into the draining lymph node, and that this event was dependent on ZEP1. Furthermore, mice which were infected with CMV could not control secondary infection with Legionella pneumophila, indicated by higher bacterial burden 6 days after bacterial infection, and they could not recover their body weight as the control group could. And this was due to reduced neutrophil recruitment caused by abolished chemokine production of CMV-infected alveolar macrophages. So in summary, respiratory tract infection with MCMV leads to a dichotomy of alveolar macrophages, non-infected bystanding and the infected alveolar macrophages. They experience an extensive loss of their immune phenotype, accompanied with increased migration and invasion, and this ultimately facilitates viral spread. Furthermore, the disruption of macrophage hallmark functions increases the susceptibility to secondary infection. And with that, I would like to thank you for your attention. Again, the DGfI for awarding me this prestigious award. Thank you very much. I'm very happy and feel very honored. The sponsor, of course, Biotest. Where's Katharina? There, yeah, thank you very much. But most importantly, the research group around Philipp Hennecke with all these wonderful people, especially Julia Kolter and Anna Löstlein, who's also presenting later today. All the collaborators, and I highlighted here Scholt Rusic, who's a fantastic scientist, virologist in Freiburg, and the funding agencies, as the Grüner Fresenius Foundation and the Medical Sciences Program. Thank you very much. Thank you.
Thank you very much for this great insight into a nutshell of your fabulous work. So now we come to the postdoctoral thesis, postdoctoral award, the Fritz und Ursula Melchers Award for postdoctoral work. The sponsors are the Ursula and Fritz Melchers Foundation, and we are very happy that Fritz is here. We have a laudatio by Stefanie Scheu, who is already here from Düsseldorf, and we are curious to hear why the committee selected our next, if you just go next, why Tim Rolensky is the one who is winning the Fritz and Ursula Melchers postdoctoral prize. Stefanie, please.
Yeah, thank you very much. I'm stepping in for Tim Sparwasser today. And also our committee had a great time and a difficult time at once to select from many excellent applications, but then we found a real worthy winner of the Fritz and Ursula Melchers Postdoc Award as Dr. Tim Rolensky for his work on antibodies of the intestinal mucosa and the mutual relationship between host and microorganisms. We were especially fascinated by the mouse model that Tim developed and used. So he basically reconstituted mice that are antibody deficient with a single strain of E. coli and could analyze the whole set of secreted antibodies of the IgA subtype and their function in vivo for the first time. And this was really a huge step forward. And also the topic fits very well with the Fritz and Ursula Melchers Prize, but this, I have to say, is not a selection criteria. And also we think that this prize also comes at a very good time for Tim to when he might want to start his own lab. So we really are looking forward to hear about your future scientific achievements. And with this, I'm handing over to Tim, and I'm also asking Fritz Melchers to the stage to hand over the deed.
We're very happy to have you here as the sponsor together with Ursula, your wife. Tim, this is the award for you. We are curious to hear about your work by yourself. This is the certificate. There is also the check. Please keep it tight. And this is also the little sign for the German Society of Immunology that we are all wearing. So congratulations to you. Congratulations also to Fritz and Ursula for this great award, because we always see that the young people are really the ones who are the next generation of immunologists, and now the stage is yours, and we will hear about your work from yourself. Thank you very much.
Yes, thank you very much. Such a great honor. I still know Fritz from my time when I did my PhD in the Max Planck in Berlin. And then Fritz was always the one that closed, after the PhD party, he closed the doors with us, basically. And in the next morning at 8:30, he was the first one sitting there asking the first question. So I think Fritz was one role model to all of us, to us young PhD students, and also one of the people that loves data the most and loves science the most I have met in my life. So it's a great honor to receive this prize. And yeah, as said, I'm interested in. Do we have a microphone? Does this work? Okay. Okay. So I'm interested in B cells and antibodies and how they work in our mucosal antibody responses. And with this, you know that we are talking about IgA because it's the most produced antibody isotype at our mucosal surfaces. And we are talking, oh, what is happening here? Okay. And we are talking about effector B cells that reside in the lamina propria, and they are secreting dimeric IgA to the intestine. And one thing that fascinates me about this system is that if you sequence those cells and you look at the repertoire, basically, you see an oligoclonal distribution. So you have large clones there that express the same or very similar antibodies. So if you could basically say that the diversity is relatively low in the system on the B cell side. But if you look at the microbiota, what you see is that it's actually quite diverse. So it's one of the antigenic, most diverse substances that we know. And can we stop this somehow that it goes on? These are not my. Yeah, okay. So this is very diverse. You have 1,000 different species. You have each of those have 5,000 different genes, and we don't know the number of epitopes basically that it has. So the idea was, how do you deal with this antigenic diversity? Is that you need to generate polyreactive antibodies that are just sticky and just binding to all these different kind of bacteria. But what we could show is that the system is actually able to mount highly antigen-specific antibodies. And we could also provide one explanation how you can target multiple different bacteria, because you could target the same epitope in different bacterial strains. So also these antibodies could be bacteria cross-reactive. So now, as was already introduced, we used a system that was already known for almost 20 years, and we used that uses blood dimeric IgA, and if you inject basically IgA, it gets translocated via the liver into the bile, and via the bile duct into the small intestinal lumen. And this looks something like this. So we use J-heavy deficient mice, and we colonize them with a single E. coli strain, and then after 3 days you inject a certain amount of IgA, and then you see a rapid decay of IgA in the serum, and you see after around 8 to 10 hours, you see a peak of IgA in the feces. So this allows you basically to reconstitute a mouse with one single monoclonal IgA antibody and study the function of it in vivo. So we did this, and we also did several in vitro assays to determine functions of IgA. These are antigen-dependent functions that are so dependent on where the antibody is actually binding, and also generic functions. So we found that it alters the nutrient uptake, it alters gene expression of the bacteria, it can impact the susceptibility of phages, also the aggregation, and also the generic mechanisms were that it can protect from bile acid-induced membrane damage and also reduce motility of the bacteria. So a single monoclonal, so then we could show that not only these components basically of the overall secretory IgA response, they have all these different functions, but you only need one antibody to induce many of those functions. So one antibody has parallel functions, how it impacts on our intestinal microbes. And at the moment, we are now going a bit deeper into this and studying a bit more about these functions. So with this, I would like to thank Andrew McPherson in Bern and the whole team, because working at germ-free level is always a team effort. You always need at least 2 pairs of hands to work with these animals. And I also want to thank my mentor, Hedda Wademann, and her whole team to supporting me throughout the last years, and also Jolanta, who is our glycan expert. Also, I want to thank you for your attention, the DGfI for the prize. And so if you're interested in this topic, we are hiring at all levels, and you can also contact me via Twitter or anything. So thank you very much, and yeah.
Smart move. So now we come to the Herbert Fisher Postdoctoral Prize for Neuroimmunology, and this is sponsored by the Rosa Laura and Hartmut Weckele Foundation, the couple in Munich who was working on neuroimmunology for decades. We are very happy that Ari Weissmann from Mainz is giving the laudatio. And Ari, I think you also had, together with the committee, a great pleasure to select from a lot of excellent applications. The winner in this is David Schaflik, and we will hear from you why the committee had the opinion that this would be the best neuroimmunology work in the last year, and this award should go to David Schaflik. Thank you.
Well, thank you, Chris, for this introduction. You took half of my speech already, but yes, it's a pleasure to present to you today the Herbert Fisher Postdoctoral Prize. Herbert Fisher was a Max Planck director in Munich, and this prize... Freiburg, sorry. Yeah, okay, sorry. In Freiburg, at least Max Planck, I was right. And this prize was initiated by the Weckele family, and it is specific to neuroimmunology. So different topics in neuroimmunology, like multiple sclerosis, which is indeed the subject of this person that got this, and this is David Schaflik. Indeed, he was among a group of excellent applicants, and we had to grade them based. Each member had to give a certain grade, and the one that got the best grade was indeed David for his work on the single cell sequencing of immune cells, and then specifically on identification of B cells in the boundary tissue of the central nervous system in patients with multiple sclerosis. This is an organ that is of high interest now in the field of neuroimmunology, the whole boundary between the central nervous system and the periphery, and how those myeloid cells and B cells and T cells that are sitting there actually affect the development of the disease. So with this, I will call David to the stage.
This is it. So congratulations. This is your certificate, and we were lucky to hear now what you are doing in addition to what Ari already told us, and why this award goes to your work. Thank you very much, and the stage is yours. Thank you.
Yeah, hello everyone, and thanks a lot for this really kind introduction by Mr. Weissmann. It's a great honor for me to be the winner of the Herbert Fisher Prize, and I also want to thank first the Rosa Laura and the Hartmut Weckele Foundation to fund this prize, and also, of course, for the DGfI committee that chose my work as the winning prize. And I will first give you just a short overview into this work that I've done over the last years, which was now awarded, or most of it. So this was published in Nature Neuroscience in July last year. And because I know that most of you are non-neuroimmunologists, I will really go quickly into it. So as Mr. Weissmann already said, there are some tissues next to the CNS which drew a lot of attention over the last years, because the CNS is here is enchathed, so surrounded by the meninges, which is one is the pia mater, which is just on top of the parenchyma. Then you see there's a subarachnoid space filled with the CSF, or also known as liquor. And then on top, just connected to the skull bone, is the dura mater. And historically, it was said that these components only support mechanical protection to the brain, so for any damage by hitting your head or something. But over the last years, there were more and more evidence and a lot of publication that showed that these tissues also might have really important immunological functions for the brain. So, as there was any deep characterization of these cells inside these tissues lacking, we thought, or my project was to systemically analyze the composition of tissue-resident leukocytes in these border compartments of rodents and also humans in health and disease. And disease, there we mostly focused on EAE and multiple sclerosis. So what we did first in the murine model is we did some CD45 IV injection to label all peripheral cells, leukocytes. Then I isolated all the different tissues that are associated with the brain, did a single cell isolation, and then I sequenced for only the true tissue-resident cells, so all those that are CD45 IV injected negative. And then we did some unbiased single-cell RNA sequencing and got these clusters, and where you can basically see almost every kind of leukocyte is somewhere present around the brain in those compartments. When you now do the comparison between the different tissues, you can see that every composition is unique. It's just every tissue seems to have different cell types in them. And because it was so many different findings, we had to focus on one finding, and that was this huge B cell population in the dura, which is completely absent in all the other surrounding tissues. And we also saw this B cell 1 population, which is not anywhere else, also not in the blood. So then we were really surprised, okay, what are these cells? We also did then some EAE mice to look if the composition in the dura mater changes, and indeed we could see that there's a lot of things going on in the dura. But we focus first on this B cell 1 cluster, where we didn't know what kind of cells these were. So we isolated these cells, did a reclustering to increase the resolution, and then we saw that this one cluster separated into 4 different clusters. When we looked at the different transcript, we were really surprised that all these transcripts indicate that in the dura they are late pro-B cells, pre-B cells, and immature B cells, which are normally only described to be found in the bone marrow and not somewhere else. So then we were really surprised, okay, is B cell hematopoiesis happening in the dura? So first we needed to check if these B cells are really found inside the dura. So I did a whole mount staining with RNA in situ hybridization. And indeed we could find precursor B cells that showed these transcripts that we already identified in single-cell RNA sequence inside the dura in wild type and also REC deficient mice. So to verify that these cells do not come from the peripheral blood, we did with some collaboration partners in Madrid, we did a parabiosis experiment where we surgically adjoined 2 mice, 1 reporter mouse, 1 multi mouse, and after 7 to 10 weeks of sharing the same bloodstream, you get in all the tissues that have a high influx of peripheral leukocytes, you get almost an equilibrium between host and donor-derived cells, so 50-50. But we could see that surprisingly in the dura, as well as in the skull bone marrow, which is expected, a lot of the cells are only host-derived, so they do not come from the blood, but are tissue-resident. When we look at the precursor B cells that we just identified, we could see that they are almost all exclusively from the host mouse. So in that way, we showed that they do not influx from the peripheral blood, but are true tissue-resident. And our conclusion was that the dura mater seems to be a place of B cell hematopoiesis, which is quite unexpected. This topic drew a lot of attention, also by other groups that did a lot of research, and there were also 2 other papers which just were published a few weeks before us, which showed by the Marco Colonna group, which showed exactly the same results as we did. And half a year later, there was another by the group of Chu from China. They also showed the same results as Colonna and us, and they additionally also could see that the B cells that are inside the dura tissue, that they seem to be primed towards tolerizing CNS-derived antigens. So this, in that way, they speculate, or because of these findings, they speculated that the B cells seem to be a reservoir that is specifically primed or specified for the CNS environment to fight infections and so on, or to tolerize also autoimmune diseases. So due to these results, there's still a lot of work to be done, and I hope I can finish with it in the next years. Yeah, so I just need to thank my colleagues and also co-authors, Jolene Warbert and Michael Heming, also my PI and group leader, Gerd Meyer-Zerhörste, our head of the institute, Heinz Wiendl, and our collaboration partner, Andres Hidalgo from Madrid. He's now at Yale, who made it possible that I could go to Madrid during Corona times and do parabiosis experiment, and Arthur Liss from Munich, and of course the whole group. Thanks a lot.
So, thank you very much for this very elegant insight into your work. Now we come to a very special award for two reasons. This is the award of Werner Müller, named after Werner Müller, for infectious diseases in developing countries. And this award is sponsored by the company Trianni, and we have Gloria Esposito here as representative from Trianni, and we're very, very happy that you're for the last 5 years sponsoring this very special award, where we always really try also to cover topics outside our own community as infectious diseases in developing countries. Now, this was like the first funding period of the Werner Müller Award, and we had so many good applications that we decided together with the laudator, Jonathan Jansch from Cologne, that we will make an exception. So we will have 2 awards of the Werner Müller Award for this year. Therefore, we will have 2 laudationes by Jonathan Jansch, and you will see that we will have 2 very, very interesting awardees. And the first is Alexandra Ehrens. And Jonathan, you will give us insights why she was the first to be selected for this award. Thank you.
Thank you, Christine. Maybe I have to thank again the sponsor for having been sponsoring this for the last 5 years. And to be honest, it wasn't very clear whether that would continue, this prize. But I'm very happy that Austriani is kicking in now and made a commitment to further sponsor the prize. So this allows me now to thank Trianni for the last 5 years, and also to thank Austriani for making this commitment, because now I don't have to give you an eulogy to the Werner Müller Prize, but instead we can talk about 2 very good applications that we had. We had many applications, and we had a hard discussion, and we ended up to split the prize. And the first awardee will be Dr. Ehrens from Bonn. She studied biomedical sciences at the Bonn-Rhein-Sieg University and did her doctorate in the Hübner Lab at the Institute of Medical Microbiology, Immunology and Parasitology in Bonn. And she has been working there as a postdoc since January 2020 and is working on the immune response against parasitic filarial nematodes. And in addition to that, and that is also very outstanding, I think, she is critically contributing to the development of new macrophyllaricidal compounds. So I think we are here to hand over the prize, which should be to you. So if you would come to the stage, please, and also maybe Werner Müller and Gloria together. Yes.
Yes, and also Werner, please come to the stage. Nee, Mensch. So. Thank you again very much. This is now your audience, and you will give us some insights on your work as the first part of these 2 prizes for the Werner Müller Award. Thank
you. Thank you. I'm not talking about malaria, though. So I think the title is wrong. So I hope I still am the prize winner. Okay, so before I start, I would like to say thank you to the DGfI and to Triani and of course Werner Müller. And I'm really honored to receive this award, and I'm happy to present my data today. So I'm working on filarial nematodes, and they can cause debilitating diseases such as onchocerciasis, also known as river blindness, as well as lymphatic filariasis, which can lead to severe lymphedema in patients. And within the NTD roadmap by the WHO, the elimination of these diseases are targeted by 2030. Now, to achieve the elimination of these diseases, we need different strategies. First of all, we need new drugs to treat filariasis, and we have to improve our knowledge on protective immunity. Now, to understand why we need new drugs, we have to look a little bit into the life cycle of the worm, which is quite complex and it involves a vector. So the current drugs that we have only target the progeny of the worm, the so-called microfilaria, but not the adult worms themselves. And this, of course, then inhibits the transmission of the disease, but the drugs have to be given for the lifespan of the adult worms, which can be up to 15 years. And therefore, we need new drugs to treat the adult worms and kill the adult worms. And these drugs are called macrophyllaricidal compounds. Now, with the help of industry partners and non-profit organizations and academia, we were able to identify several different of these compounds. And two of them, ABB4083 and imodapside, are currently in phase 2 studies. And then we have two additional drugs, including oxbenzazole, which are in phase 1 study. And then we have two further drugs that are under preparation for phase 1 study. Including corallopyridine A, which has been developed in our institute with the help of the DGfI. And moreover, we have additional drugs that are candidates on hold, but which can also be used to treat filarial diseases. Now, in addition, we also have to improve our knowledge on protective immunity to identify, for example, new targets for the treatment. And when we look at the immune response towards filaria, it's quite striking that we have an accumulation of eosinophils during this infection. And eosinophilia is even a hallmark of filarial infection. And when using rodent models and knockout mice, so a rodent filarial model, we were able to show that eosinophils mediate protection against filaria. However, we don't know exact which mechanism they use. And eosinophils have different effector mechanisms, including the etosis, where intracellular DNA is explosively released into the surrounding environment, which can capture especially large pathogens such as filaria. And now to investigate if eosinophil etosis contributes to the protection against filaria, we generated eosinophils from the bone marrow of mice and co-cultured them with microfilaria, so the progeny state of a rodent filarial nematode. And we were able to see that these microfilaria induced DNA traps in the eosinophils. And next, we then wanted to investigate which pattern recognition receptor is involved in detecting the microfilaria, leading to the release of the DNA. And therefore, we performed again co-cultures and added blocking antibodies to this. And we demonstrated that microfilaria significantly increased the DNA release by eosinophils, which was significantly reduced when a dectin-1 blocking antibody was added, but not when we blocked dectin-2, Minkel, or when we used an isotype control. So the MF-induced eosinophil etosis is dectin-1 dependent. And finally, we wanted to investigate the in vivo role of these traps. And therefore, we co-cultured microfilaria with eosinophils in the absence and presence of DNase, which destroys the DNA traps. And then we injected these microfilaria intravenously into mice. and we demonstrate that the microfilaria are cleared from the peripheral blood over time. However, this was significantly enhanced when the microfilaria were covered by DNA traps. And thus these DNA traps enhance the microfilaria clearance from the peripheral blood. Now, this was a really, really brief overview of my work, so I've done much more, but if you're interested to hear more about eosinophilatosis in response to filaria, come visit me tomorrow during the second poster session. I will be standing next to poster 75, and I'm looking forward to see you there. Yeah.
So without further ado, Jonathan, this is your second term. There's a second laudatio, and we are very curious to hear who will be the second winner. And those of you who are standing here in the back, please just come down now. This is the best moment to find your seat, because you certainly want to have a seat instead of standing there in the floor. So thank you, Jonathan. This is your second laudatio.
So the second laudatio will be on Dr. Ann-Kathrin Lößlein, and she studied human medicine at the University of Freiburg. And during that time, she was also a fellow of the Deutsche Studienstiftung. For her doctorate thesis in the Hennecke laboratory, she was supported by the Freiburg Motivate program of the Else Kröner-Fresenius Foundation. And her doctoral thesis was awarded with summa cum laude. And since 2021, she's also funded by a clinician scientist program, IMPACT, which is also sponsored from the German Research Foundation. Dr. Anne-Kathrin Lößlein made an outstanding contribution in our understanding of innate immune responses in the defense against mycobacteria that, as we all know, can cause tuberculosis. And therefore, she will awarded the Werner Müller Prize for her work on the development of giant cells from monocyte precursors in tuberculosis. So congratulations, Dr. Lößlein, and I think, may come to the stage so that our president can hand over, together with Gloria, the prize to you.
Yes, exactly. So, this is your certificate. Gloria Esposito is again here on stage. Werner, can you do me a favor and just wave your hands? He's sitting there, by the way. So this is yours. Thank you very much, and we are very curious to hear why you are receiving this award. Thank you. So the stage is yours.
Thank you very much for this very nice introduction. And first of all, I really want to thank the German Society for Immunology, the award committee, of course, Professor Werner Müller, and the sponsor, Triani Inc., for awarding me the Werner Müller Prize about my work on monocyte progenitor cells, which can give rise to multinucleated giant cells. And I now have the chance to give you a short introduction to this project. So we are interested in mycobacterial infections, and tuberculosis is an example of an infectious disease mainly caused by Mycobacterium tuberculosis. And there is very early evidence of TB in humans, as you can see here in this example of a skull of a Homo erectus with signs of leptomeningitis tuberculosa. TB was a major health burden during human history, and from this the question arises if it's still relevant today. I'm sorry, there seem to be some layout changes. We can answer this question with a clear yes, as it's one of the top 10 causes of death in low-income countries, and around 25% of the world's population is infected. But interestingly, only around 10% develop active disease, so we have a lot of latent infections. And the transmission happens via an airborne route, and then the mycobacteria can induce in the tissue granulomatous inflammation. And these granulomas are very complex immune cell structures containing many different immune cells, especially macrophages. And here you can see an example of an immunohistochemistry staining of a CD68 positive glycogranuloma, which we induced with BCG. And this is Bacillus calmette-guerin, the vaccination strain also belonging to mycobacterial tuberculosis complex. And hallmark of the granuloma is the multinucleated giant cell. And this is a specialized macrophage which can keep mycobacteria over very long periods. They contain at least 3 nuclei and can produce nitric oxide. So in this project, we wondered which role monocyte progenitus play for mycobacterial infections and these complex immune cell structures, and if multinucleated giant cells are the progeny of a specific subset and how they develop. And therefore, we analyzed different monocyte progenitors, and due to time reasons, I want to cut this short. We mainly focus on common monocyte progenitors, and they are restricted to the formation of mature monocytes. And when we compared the potential of these cells to form multinucleated giant cells, we found that especially progenitor cells, so these common monocyte progenitors, have a high potential to form multinucleated giant cells. And if we analyzed the transformation process, we found an accumulation of lipids in these cells. And this accumulation was accompanied by the downregulation of cholesterol efflux pumps, which are located at the cell membrane. And this significant downregulation in infection also supports the accumulation of lipids in the cell. Additionally, we were able to alter giant cell formation by altering lipid metabolism, and therefore we added, for example, methyl beta-cyclodextrin to extract the cholesterol from the cell. And we could reverse this effect by adding LDL particles and promoting the uptake of cholesterol to the cell, which induced again giant cell formation. So it seems that lipid metabolism is an essential prerequisite for giant cell formation from progenitors. And of course, we further wanted to characterize the role of monocyte progenitors for mycobacterial infections. And therefore, we went into an in vivo model and worked together with Anka Dorhoy and Lisa Scheuermann from the MPI in Berlin. And they performed an aerosol infection with Mycobacterium tuberculosis. And when we analyzed bone marrow and blood of these mice, we could identify a new progenitor subset, which we called induced monocyte progenitors. They are more mature than CMOP, but still do not express CD11b like monocytes. And interestingly, and in contrast to other progenitors, we can find these cells in the circulation in mycobacterial infection. And to further characterize their function in granuloma formation, we went in our BCG intravenous infection model, where we have granuloma formation in liver and spleen. And here we performed an adaptive transfer of progenitor cells. And what we observed was that the progenitor cells, which you can see here in green, can contribute to granuloma formation in vivo. And finally, we wanted to analyze the role of progenitors for multinucleated giant cell formation. And therefore, we worked together with A.G. Hölscher from Borstel and used the IL-13 transgenic mice. And they are very susceptible to mycobacteria and can form mature granulomas. And in this model, we can also analyze giant cell formation. So here we performed, after MTB infection, an intratracheal transfer of progenitors. And indeed, we could observe that the transferred GFP positive cells form part of nitric oxide producing granuloma-like structures, and we have atypical nuclei and cells with at least 3 nuclei. So we have multinucleated giant cell formation from these cells. And with this short overview, I would like to come to my conclusion. So we could show that progenitor cells have a very high potential to form giant cells, and that the lipid metabolism is an essential prerequisite for the transformation, and that we could identify a new progenitor subset which can circulate in mycobacterial infection, and that these progenitor cells can contribute to granuloma macrophages. And with this, I would really like to thank you for your attention, and my PI and supervisor, Professor Philipp Hennecke, for his fantastic support during the last years. The whole AG Hennecke, especially the people who contributed to the data, all our cooperation partners, and of course the Motivate program and Impact program, which Professor Jansch already introduced. Thank you very much.
So thank you very much for these 2 wonderful talks on infectious diseases in developing countries. And now we come to the George Kohler Prize for young immunologists who have just started their independent laboratory and are creating their own career with their own topics coming out of their postdoctoral phase. This is named after George Köhler, and we're very happy that Christian Bogdan from Erlangen will give the laudatio, but I have to admit that Maike Hoffmann unfortunately is sick, so she will not be able to be here, but we thought Christian would be the best person to tell you why she was selected for this award, and then we will hand over her certificate and everything that comes together with it as soon as we see her, or we send it by post. But Christian, first, the stage is yours, and we're curious to hear why Maike Hoffmann was the choice for this prestigious award. Thank you.
Yeah, thank you, Christine. First of all, I have to say I have stepped in here for Yvonne Sonntag, who cannot attend this meeting, and normally I'm more in charge of the Robert Koch Postdoc Prize, but I'm very happy to talk about the George Köhler Prize. For the young guys in the audience, George Köhler is one of Germany's Nobel Prize winners who used to work in the Basel Institute of Immunology in England, at the Max Planck Institute in Freiburg, and who won the Nobel Prize together with Niels Jerne and Cesar Milstein in 1984. And the George Köhler Prize actually was introduced in 1998, and this was actually meant to be given to young investigators who have already established an own group. So this whole ceremony here is like in the RD TV Awards. So we are stepping up in the career, and now we are in the young or middle-aged group of people who have established their own laboratory and who have been working for quite a while in a particular area of research. So Maike Hoffmann cannot be here, but I will briefly tell you what she has been doing. She had studied molecular medicine at the University of Freiburg. So some of you might notice that we have a strong bias towards Freiburg here in this ceremony here, because it's now number 3 and it's not the end yet. And after her molecular medicine studies in Freiburg, she joined the laboratory of Hans-Peter Pircher at the Institute of Medical Microbiology at Hygiene, where she in fact started to work on CD8 positive T lymphocytes. And during her diploma thesis, as well as during the subsequent PhD thesis, which she was also doing in Hans-Peter Pircher's lab, she already focused on the characterization of CD8 positive T cell subtypes. She was working on E-caterin and KLRG1 as marker molecules. After that, she became a postdoc in Robert Timmer's laboratory at the Medical Clinic number 2 in Freiburg. And there she actually took her knowledge of CD8 positive T cells in order to apply it in hepatitis B and C virus infections. And one particularly important observation she actually made was that there are 2 different categories of CD8 positive T cells. There are the classical, of course, effector T cells, there are memory T cells, but there are also exhausted T cells. And she could show that exhausted CD8 positive T cells are very much dependent on persistence of antigen, so that people who get treated for hepatitis B and C, they actually then have a decrease of these exhausted CD8 positive T cell population. In 2016, she became then at the same clinic a junior research group leader, acquired really quite a number of tertiary money projects, is members of SFBs, and continued to work on the characterization of CD8 positive T cells in viral infections. And most recently, she also used her really great knowledge on the characterization of these cells in the context of SARS-CoV-2 infections. Her publication record is really outstanding. It started already with the diploma and PhD thesis with a PNAS paper, and then she climbed up the whole Nature categories of journal. So Nature Communications, Nature Immunology, Nature Medicine, and most recently Nature paper where she could show that CD8 positive T cell responses are actually preceding the development of memory B cell or B cell formation in the context of SARS-CoV-2 infections. So I guess that's what I can tell you, more or less out of my memory. It's unfortunate that she's not here. The prize is sponsored by Biotest again, so we are very grateful to the Biotest company that this is the case. And if you have any questions on Maike, then you just have to write her an email. I guess she will respond. She's a very kind person, And one of the prime examples that you can be extremely successful even if you stay at one and the same place in science.
Thank you very much. So, thank you very much, Christian, for stepping in the first place, for Yvonne, and then also explaining why Maike Hoffmann was selected for this award. And now we come to the highest award that we do have in the German Society of Immunology, and this is the German Immunology Award. This is sponsored since a couple of years by the company AbbVie, and we are very happy that Stefanie Leue is here, and she will be there on stage then to just sort of be part of the certificate handover for this award. You may have realized that we have in this year the two awards together for the year 2020 and the year 2022, because we thought in our virtual event last year it would be really a shame if we would not be able to have Thomas Boehm here as a speaker and as the sort of winner of the German Immunology Prize by himself in his actual, oh, you guys just show this, sorry. So here we are. And you see here that we have former laureates for this prestigious award, and now we are coming to Thomas Boehm. And we are very happy to have you here, that you will get now a little later than planned due to pandemic your award. And Reinhold Förster, this is now your stage, and you will give us the insight of our selection for Thomas Boehm. And I think this shows how broad immunology is. We will go now into the deep molecular mechanisms of evolution. And then we go all the way down to translation. And this is what we are all about in immunology. And that's why I really like it that the both awards will be here in the ceremony for 2020 and 2022. Reinhold, please.
Thanks very much, Christine. Actually, I thought we have changed the program, but it's good that we stay. It's all super. Thomas Boehm is a true immunobiologist. His research focuses on the development of the immune system in individuals, but also during evolution. He elucidated several mechanisms of T-cell development, and he identified the molecular basis of immunodeficiencies. So he and his team were able to elucidate the basic mechanisms of T-cell development, and they identified genes that are essentially involved in that. For example, they found out that the FOXN1 gene is indispensable for thymic T-cell, for thymus development in all vertebrates. But he also described the molecular basis of various immune deficiencies, and he was also able to identify the first oncogenes in T-cell leukemias, and that was quite a while ago. He does not only want to identify, prescribe the mechanisms, but he also tries to find ways how to correct them. For example, he tries to aim to establish an artificial thymus in a living organism, in a mouse, with the aim to re-regulate dysregulated T-cell development. He investigates the immune system, and that is very particular for him, not only in the mouse or in humans, but he investigates them comparatively in many species. And that are mice, that are zebrafish, it's sharks, and it's lamprey. And in particular, the latter one is, of course, very interesting in terms of science, because those jawless fish separated several hundred millions of years ago, and they used different mechanisms. But still, how the immune system works, how we can get gene rearrangement and also a variety of genes, that is also maintained. And that's why his work is so unique compared to many of us. Thomas studied medicine in Frankfurt, and he also had some study visits at the Columbia University and at the Royal Marsden Hospital in London. In 1982, and if you would look at him, you would not believe that this is already 40 years ago, he got his doctorate in Frankfurt, and 6 years later, he also was getting his habilitation at that place. He then went for 5 years to the MRC in Cambridge, and when he came back, He was appointed Professor of Molecular Biology at the University of Freiburg and Professor of Experimental Therapy at the German Cancer Research Center in Heidelberg. Since January 1998, he is the director of the research unit Development of the Immune System, and again, it's Freiburg and not Munich, and there he is leading a group studying or doing all that stuff what I've been describing before. Thomas Boehm is elected member of various scientific societies, including the European Molecular Biology Organization, the National Academy of Science, Leopoldina, and the Heidelberg Academy of Sciences. Since 4 years, he also took chairmanship of the Board of Trustees of the Paul Ehrlich Foundation. The work of Thomas Boehm has been honored with numerous awards, including the Gottfried Wilhelm Leibniz Prize, the Ernst Young Prize, and as one of very few scientists so far all over, he got awarded 2 ERC advance grants from the European Research Foundation. Today, this list gets another entry, the German Immunology Prize 2020. As Christina said, better late than never.
Thomas. So, Thomas, here, this is your certificate. Thank you very much for coming. And as Reinhold said, this is really something we are all looking forward to, because this is a masterpiece of immunology that we will hear now from your presentation. And we will then celebrate your award for the German Immunology Prize. Thomas, bitte.
Yeah, good afternoon. I'm greatly honored to receive that prize. You can see I looked much younger 2 years ago, so something must have happened. Actually, I have to tell you, it was a bit of a shock when I received an email by Thomas Kamrat at the time, and the email was dated April 1st. And I thought, okay, well, typical mistake, hoax. But then I became a bit curious, so I telephoned, and it turned out to be true. So thank you very much to the Society for awarding me this wonderful award. And thank you, Reinhold, for this very kind introduction. Greatly honored. So what I would like to do is I would like to give you I have only 2 hours. I will give you, discuss just one project, which I think is a bit out of the mainstream, but shows what one can do when one looks into the diversity of nature and really lets Mother Nature tell us what she can do. So it's about evolutionary novelties in the vertebrate immune system, and we can begin by showing you this slide of my group of people. A real exciting and dedicated lot, a fearless group of scientists who really go with me on these journeys, often long-winded, but sometimes we come up with some interesting results. The work I will be discussing was led by Jeremy Swan. You see him here indicated with the white name. You also see Hans-Peter Pircher to the right. He joined my laboratory after he retired from the University of Freiburg, so this is another Freiburgian which should be named here. Okay, so what is this all about? You may know that adaptive immunity, that is the facility of somatic diversification of antigen receptors during the lifetime of an individual, was invented twice at the early days of vertebrate evolution, about 500 million years ago, and we have 2 branches that survive to this day, the jawless vertebrates and the jawed vertebrates. The jawed vertebrates is our group, about 60,000 species, and the jawless vertebrates didn't do so well. There's only about 200 species left. Nonetheless, you see many failed attempts, and we tend to think, of course, that these failed attempts are due to the fact that they didn't manage to get adaptive immunity off the ground. So this gives us a lot of room for comparative analysis. So we have this split occurred 500 million years ago. So, of course, these animals that we study today didn't live from that time, but certainly this split gives us a lot of information. The important point I would like to make today is, when you think about it, is that at this time when adaptive immunity was invented, it started co-evolution of somehow connecting functionally a primordial innate immune system that comes in various forms in invertebrates, and then this must have been integrated into the new system that also had adaptive facilities. So this is indicated in this little cartoon here. And so since 500 million years, there's a lot of co-evolution going on, and of course you know what the result of co-evolution is, that neither system can operate on its own. One is dependent on the other, and we see this, of course, when one looks at inborn errors of immunity, that one system cannot compensate for the other. And that may or may not lead to fatal or very severe immunodeficiency. So this is the consequence of co-evolution. And we also tend to think, this is another paradigm, we also tend to think once co-evolution has started, it becomes very difficult to go backwards. So evolution and co-evolution seems to be going only in one direction. Now, but is this actually true? Is it perhaps possible that co-evolution can be reversed? Because that would make a very interesting outcome, because one can then look at the disentangled immune system, probably with a new rearrangement or new arrangement of immune facilities. So this project started, not from my lab, of course, but this project started 100 years ago with an observation made by this man, Tate Regan, here pictured when he was a student at Cambridge. And he described in the Proceedings of the Royal Society in 1925 a particular observation that he discovered as keeper of zoology. He had plenty of time actually spending time in the dark cellars of the British Museum. He found a particular class of deep sea fishes where male fishes were fused permanently to the body of females. And this is the original publication that was, as I said, published almost 100 years ago. And when you start reading old papers, even papers that are in the century before, so from the 1980s, you will find super interesting things, and this is one of them. And here is what he described a little later, this sexual parasitism. We heard about parabiosis these days. This is effectively the same thing. The male attaches to the female. They form one chimeric organism. The male reduces itself over time. It basically leaves only the testicle, because that's the only important bit for a male. So when the female is ready to spawn, the male releases the sperm, and in this close proximity, fertilization happens in the abyss of the deep sea, and the fertilization then hopefully is successful, and the next generation of fish can come. But the important point for the immunologist's point of view is that they have an intercommunicating blood system. This is basically what we do artificially when we do parabiosis, but this is what they do naturally. Now, when you think about this, it seems a bit strange. How is that actually possible? Because we know that when we do this artificially, they have to be genetically identical. We can do this with genetically identical twins, and experimentally with genetically identical animals, but this is a rather strange thing. So, how is that possible? A summary of what was known until 2009 about this strange phenomenon in nature is published in this book, and I show it here. It makes interesting reading. They look a bit ugly, these fish, as you can see, but Theodore Peach was a key person in driving this project, because he is the godfather of this particular area of science, and he opened all the doors for us, for all the natural history museums in the world, so they would spare us a little bit of material so we could study the genome of these particular fishes. So this is what he collected and what he composed. This is the 4 types of anglerfishes that one can distinguish. There are those that have no attachment, so where male and female live freely and they come together from time to time. Then there is attachment on a temporary time scale, where the male attaches to the female temporarily, spawning occurs, and the male goes its own ways again. Then there is permanent attachment in a one-to-one mode, as we call it, where there is typically one male attaching to one female. And there are the greedy females in the permanent multiple attachments, where one female has many males. Can't have enough, just has many. And you can see the world record here is one female with 8 males attached to it, or to her rather. So Jeremy went about trying to collect all this material. It took us about 10 years to get the material together, and particularly we were interested, of course, in having these parasitized pairs, a female and a male, so we could really study the genome and the immunogenome of male and female in these parasitized constellations. And I must stress, there is no living animal that we can study. It's not possible to do this. The museum curators would only give us a tiny speck of skin, because they would not destroy their specimen. So the only thing we can do is we can extract DNA from essentially nothing, and then do a genome sequence and then analyze the genome. There's not even RNA. So this is really a very difficult project, but Jeremy did a very wonderful job. So the simplest explanation, of course, is that they are clonal species. We know of examples. Your flower pot snakes are clonal, and they colonize the entire world. So it's possible that they might be clonal. That would be a rather boring outcome, unfortunately, or fortunately for them. This is not true. When we do this genome, each individual is really different. There's an enormous amount of heterozygosity, so clonal species is out. Then they might perhaps assess MHC status before they fuse, like we do in the laboratory when we do organ transplantation, because we know that fish and other animals can sniff out MHC haplotypes by their olfactory facilities. So we looked at this particular possibility. Again, this is out because fused males and females do not share MHC alleles. So there is no match that arises from whatever mechanism. This is not the case. So the two most plausible things were out. So what is left? Maybe that these species somehow manipulated the immunogenome such that this fusion is easily possible. So, but how does that work? So I'll go through the 3 groups of fish that I explained, the temporary attachers, the one-to-ones, and the one-to-many, and you will see how they managed to do it. Okay, so we looked at the genetic complement of genes where we can really assign definitive functions, not these pleiotropic transcription factors. This is all super complicated, but we go for really immunologically important genes. So AID, for example, important for antibody affinity maturation. So you see that here. I show you the controls, the never attachers, and the 3 temporary attaching species that we analyzed. They don't have a functional AID gene, so they cannot carry out antibody affinity maturation. And we interpret that to mean that this reduces alloimmunization, which we know from human studies is important in late organ rejection. So you see the color coding at the bottom. So green and yellow basically means okay, and then blue and red is problematic. So now we look at the one-to-one permanent attachers. Still, they don't have AID. AID is gone. What they also have is they lost one component of the CD3 complex, so we assume that TCR signaling is reduced, and they don't have CDA co-receptors. So which could mean, although we cannot do functional analysis, of course, but that could mean that they have lost cytotoxicity. That is an interesting piece of information, because we know that cytotoxicity, of course, is involved in acute rejection when organ transplantation is performed. And the extreme case is this. I still remember when I looked at one of these genomes in the evening, I just couldn't believe it. Almost a shock as this letter from the German Society. there were 2 species that didn't have functional RAG genes anymore. You can see that down there. One representative, P. spinoceps, has destroyed the RAG1 and the RAG2 genes, which are normally joined together in the genome. And ironically, these genes are destroyed by the invasion of transposons. And we know from work of David Schatz and others that RAG originates as a transposon, a domesticated transposon turned into a recombinase. And now the empire strikes back and the transposons kill the RAG genes. But that, of course, means RAG genes are gone, no antibody, no TCR rearrangement is possible. So they basically have lost what we call canonical adaptive immunity. And as a variation on the theme, you see this in the third one, and in this where these 2 blue arrows are, there is a Cryptospira coesi. They still have B cells, consequently they still have the RAGs, but they don't have T cells based on the immunogenotype. So life is possible without T cells. I never thought that would be possible. But life is also possible without what we call canonical adaptive immunity. So we know, based on phylogenetic analyses, without going into the detail because of time, this must have been invented more than once. And it originated about 100 million years ago. So these creatures now happily live down in the deep sea in pitch darkness for about 100 million years and happily reproduce with a non-canonical immune system, no adaptive immunity as we know it. So what that means is it is indeed possible to disentangle this co-evolutionary trajectory of innate and adaptive immunity. And how do they survive? How do they live? And I call this innate immune system 3.0. Because the 1.0 is the one that is before the invention of adaptive immunity. Then, of course, comes this coevolution. So we call this collectively 2.0. And now they've gone back again, taken out canonical adaptive immunity. They perhaps retain some adaptive functions. We don't know. We call this 3.0. And I think that is a rather amazing result. And it will now give us the opportunity to really study selective degradation of immune pathways, because we have now non-T cell, but B cell plus, and non-T, non-B vertebrates. And we can simply look at the various genes. This is simply here a cartoon, just to illustrate the point, that we can now go in and study the gene complement. Is this gene present in this particular genotype or in this particular genotype? And then we can build, it's kind of a genome-wide evolutionary validated genetic screen, if you wish. We can now go and look at these individual genes and then coordinate and apply them and assign them to these different pathways. I think this is a rather interesting new discovery tool that nature has provided us with. But once again, of course, this is not possible without collaborators and what I call the home team leaders. And in this case, it's relatively easy. It's Ted Peach from the University of Washington, who gave us a lot of material from his own archives and opened the doors, as I said. And Jeremy Swan, senior member of the lab, was leading this project. And you see down there the funders that are generous enough to allow us to go on these journeys. And should you be interested to come and join us on these rather exotic expeditions, we have vacancies for postdoctoral fellows. And once again, thank you very much for giving me this award. It's a very, very great honor. And thank you for your attention.
No questions.
No
questions today because we have a very tight schedule. If you want to ask Thomas, he will be around, and I think there are a lot of questions, especially on the fish kind of thing and the fusion of the male and the female fish. And now we come to the German Immunology Prize 2022. And when you've seen the program, you may have thought, well, this is sort of a no-brainer that we give it to Özlem Türeci, Uğur Şahin, and Katalin Karikó, but actually it was not. We had intense discussions, and we are very happy again that the sponsor AbbVie with Stephanie Loy, who I will ask you on stage later on, because I forgot that in the first place, supported that. And we had it took us less than 2 weeks to get this under control last year, because we thought we should do this, but not specifically for the SARS-CoV-2 vaccination, but for the 20 years of work before in the field of tumor vaccination and the development of mRNA for vaccine strategies. And so I know that you have to leave, and this is why I keep this laudatio very short, because we would like to hear from you how your impression is on these developments of the last 25 years. But I briefly would like to start with Katalin Karikó in the order of the presentations you will hear in a minute. Katalin, you said this afternoon that you were interested to really bring mRNA into the clinical application. And the original application you were interested, as were Özlem Türeci and Uğur Şahin, was the cancer therapy and an individualized tumor therapy where you select individual targets, you use mRNA techniques to vaccinate cancer patients. And so you would say, well, you just take mRNA and then that's it. So this was not it. It took 20 years, and Katalin was the one saying that if the mRNA is too immunogenic, it's not going to work. And you insisted in modification of the mRNA to make it stable, useful, and exactly the right balance between immunogenicity and the function to make the protein without being too immunogenic so that it would not work. Coming from Szeged and working at the Pennsylvania University for more than 24 years, you joined BioNTech 9 years ago. And you mentioned this afternoon because you wanted to bring it to the clinic. There was no idea that the pandemic would come. And from the cancer topic, it will all of a sudden be a race to develop a vaccine against the SARS-CoV-2 virus and the spike protein. Özlem and Uğur studied medicine in Cologne, went then to Hamburg Saar, to Michael Freundschuh's lab, and then went to Mainz, to Christoph Huber's institute when he was still the head of internal medicine at the Gutenberg University in Mainz. This is the time when we met because we shared the interest of why the hell do T cells in the tumor not recognize the tumor. So we tried to isolate the T cells and see what was wrong with those T cells and how could we improve the T cell recognition of tumor cells. So coming from your medical background, you became not only interested in the techniques, how we could improve T-cell recognition of tumor cells, you also developed the mRNA strategy as a technique where you could do both. You could go for more specificity because you could sequence the tumor in the first place, by in silico identification, generate the targets, and then generate personalized mRNA therapies for cancer patients. And there are several entities, and you will talk about that, where you have already entered phase 1 and phase 2, like melanoma, gastric cancer, but also colon cancer and others. And then it's not only the skills of tumor immunology. At the same time, what's to me pretty unique is your skills for venture capital and startup companies, because you had Ganymede in the first place, where Christoph Huber again was very instrumental to have a startup outreach of your scientific work, because it's clear that you cannot reach something like that only with academia. So then after Ganymede was sort of the first startup, and Tron came as a second consortium in Mainz, then the BioNTech company was founded, and this was then something where you still continued on the cancer field. And then there's a very interesting book where you describe the moment, and Uğur mentioned that, when you saw the sequence of the spike, and when it came to your mind that this is the antigen for a vaccine, where you don't have to do that personalized, it's only one spike. Now there are a couple more, but it was only one spike at the time. And then the Operation Lightspeed, the Project Lightspeed, was your decision to go into the race who would be winning. So the three of you were actually the driving forces behind the development. And this is why we thought we'd take this moment to say this would never, ever have been possible without the 20 years of basic work up front, because then it was able to do that fast, but not without the 20 years of development and constantly improvement of mRNA techniques. And the three of you were actually instrumental for that. So this was sort of my little introduction. And Katalin, I would like to ask you first, and then Özlem Türeci, and then Uğur Şahin here on stage, that you get your certificates. And also Stephanie Loy, please come up. And we just do that now.
forward. This. Oops. Oops. Oh. Okay, that was the 5 minutes, my talk. Where is it?
You want to go back?
Back, yeah. Mm-hmm.
Super.
Okay. Anyway, I would thank you very much. It is a real honor to receive this award and a privilege to receive this award with my fellow awardees today, and thinking about all of those other awardees who get this award in prior years. This is a real privilege. Thank you very much. I would love to talk a little bit earlier, then it would mean that I get the Young Investigator Award, but this is already gone, and today I talk about developing mRNA for therapy. RNA it started with discovery of mRNA in 1961, and it took 60 years to get the first mRNA FDA-approved product in the form of COVID-19 LNP mRNA vaccine. And during this year, 60 years, a lot of things happened. Some major things I will point out. Most of the time it is just incremental improvement of the RNA. So it started with discovering RNA 1961, and with this one we learned that the information flow in every free living organism is DNA, RNA, mRNA, and protein. Of course, the scientists at 1960 and the beginning, they couldn't make RNA, just we are doing for the vaccine. Of course, first they just could isolate. And most of the study was done with viral RNA, or they discovered that if they take a reticulocyte and isolate RNA, one type of RNA is enriched, and the scientists already started in the 60s, 70s, try to translate. So those isolated RNA, which was globin specific, they put in lysate, injected oocyte, and one interesting study was when they put in liposome, and this was 1978. And the RNA, this was a globin RNA, was put in liposome and fused to primary cells and demonstrated that the encoded protein was made. So I find this a very important finding. Of course, in 1984 and 1985, scientists introduced the in vitro transcription of the RNA, and this was Harvard University, Douglas Melton, Paul Krieg, Tom Maniatis, and many. I have no time to tell that hundreds of scientists participated in all of these projects. Even the discovery of messenger RNA was prior to 61, already 58, they already doing and try to discover, but discover the ribosomal RNA, the tRNA, and others. So what these scientists did, they realized that the phage RNA polymerase has high fidelity to transcribe from a phage promoter, which was, in that case, SP6 or T7 is a very short one, so it's very good, and you get a lot of RNA if you linearize this plasmid. And first what they did, actually, they did human beta interferon RNA they transcribed, and they just needed the template, they needed the 4 basic nucleotide triphosphate enzyme, and they also get some capping enzyme from Bernard Moss from NIH, and they made this RNA. And when they injected to the frog oocyte, and they cultured those oocytes and found that the supernatant of this medium was actually functional, so it was inhibited the viral infection. So in vitro transcribed RNA coded for a functional protein 1984. But we had to still work on. Because what happened with this discovery, people started to make RNA, which they and injected to in animals from 90, 92, 93. So first, like 92 was one paper when vasopressin mRNA was injected to rats and then treated the disease. That inspired me for many, many years. I have to say that most of this group never ever published again. I keep looking, and at the beginning was not that easy as in these days, but I couldn't find, further publication, and why most of these scientists like doing, influenza vaccine, like the Martinon team first time, and there were no follow-up. What is that? What was the reason? Because the RNA had one thing, problem because very small amount, short amount of time, very small amount of protein was made, and even this protein, RNA was immunogenic. So RNA degraded. You have to figure out how to wrap it, how to deliver. So a lot of things needed to do. And that's what happened during those 60 years. Incremental improvement of the RNA, figuring out formulation, and then sometimes the two is coming together, better RNA, better formulation. And this is most of this incremental increase. I myself, I started to work in this laboratory at University of Pennsylvania. I spent here 17 years. I did all of this RNA on this bench here, and from 89. And, it is a neurosurgery, which is just, an escape place to do all of this basic research. And at the beginning, we did a urokinase receptor encoding RNA, and the functional protein was made, which encouraged us, that maybe RNA, in vitro transcribed RNA, is good for maybe cell therapy. Sorry that I did not wear gloves and coat, and usually this is how I work. But anyway, I was doing all of these research, and one day, after working already 10 years with in vitro transcribed RNA, meeting Drew Weissman, we learned that the mRNA I am making actually is immunogenic. As a lot of inflammatory cytokines, TNF-alpha was made, and I thought that what I make RNA is just as good as inside in our bodies. And to understand why it is immunogenic, we decided and we were thinking that maybe if we took out the RNA even from our own cells, maybe, and we put to the dendritic cells, human dendritic cells, what he was working, maybe those are also immunogenic. To test out, we isolated RNA from a different compartment, and we had nuclear RNA, we have mitochondrial RNA from the platelets. That's a lot of work, and when the RNA I made, and we measured on dendritic cells, a lot of TNF-alpha was made. This purple is my in vitro transcribed RNA, and those others was not as much. And this tRNA did not induce anything. So this tRNA was not immunogenic. And knowing that tRNA has a lot of nucleoside modification gave the idea that could it be that the nucleoside modification is suppressing immunogenicity of the RNA? But of course you can say because it was shorter, it didn't have different ending, 5 primes, 3 primes. So a lot of things is in your mind, but I don't have time. So just I say, oh, because nucleoside modified, so probably this is the reason, but we have to prove that. And, and to prove that, I'm just showing you that if you fall asleep, that you see that this is complexity of modification, nucleoside modification, more than 100 different ones. And at that time, I'm talking about 2003, 2002, even the enzymes were not known what introducing these modification. So what happened, you just couldn't order one from Promega and try to modify your RNA, so we had to do differently. We have to purchase already modified nucleoside triphosphate and try to cross our fingers that the T7 polymerase will incorporate. Of course, here I'm showing 10 of those naturally occurring nucleoside triphosphate, and only these 5 incorporated by T7 polymerase. But this is how we could make nucleoside modified RNA. And then we tested out similarly as we did before, and transfected to dendritic cells and measured inflammation. And when we looked at there, the blue is that the in vitro transcribed RNA, which was not modified, but what we found is some of the modified one is not induced any inflammatory molecules. When we made a closer look, and we found that all of those, the uridine was modified. These were the uridine modified. mRNA was not inducing inflammation, no TNF-alpha. So far so good, but we want to make, or I want to make, the mRNA for therapy, so it has to be translated. So as you can see, some is not translated, like S to U, but luckily the pseudouridine containing RNA, we found 10 times more protein was produced. So that was the icing on the cake. I couldn't believe that, so I can go back and use RNA for therapeutic purposes. So we started in injecting with the RNA, erythropoietin coding RNA, and those are, generating red blood cells. If you follow the bicycle sport, you heard already about that. And what happened is that when we did inject it to the mice, this pseudouridine containing EPO coding RNA, we could see a couple of days, we could detect the protein, which has about like 2 hours half-life. And we could see also that single injection resulted hematocrit increase, so the translated protein was functional, and we could maintain, if we weekly injected, we could maintain high hematocrit in the animals, and most importantly, it was not inflammatory. So this was the time that the university thought that that's enough for me, because I didn't get grants, and but luckily I learned and that there is a company, BioNTech, and so in 2013 I joined forces with Uğur Şahin and Özlem Türeci, and we didn't have this nice building at that time yet. We were on the campus, and what we did together is further improving and implementing different kind of construct improvement. We codon optimization, purification. We introduced new UTRs and the modification. It was there, and so what we could achieve eventually is that this little problematic little protein short time. Now the RNA was prime time because we could generate high level of protein from those RNA which had a new cap. All of this part of the mRNA was improved, and so the RNA was ready for prime time. And I mentioned here 2 examples. One of them, what we started to make bispecific antibody encoding mRNA and injected to mice and demonstrated that weekly injection of this mRNA, this coding for the bispecific antibody, we could eliminate large tumor in animals. And we also did a study where, again, the messenger RNA in this cancer model is not a vaccine, because the messenger RNA was coding for cytokines, and these mRNA were injected to the tumor close to the surface, and making the cold tumor hot, and helping the immune cells to recognize the tumor, and then when they circulated, they eliminated metastatic tumors. And this actually went on human trial. So with that, I would like to thanks to my colleagues who helped me on this journey, and they wear gloves and other, coats, not unlike me. And I would also like to thank to my family. My daughter, I like to brag, my daughter is a 2 times Olympic champion, and no, because I was known for University of Pennsylvania, and they always introduce me, meet Kati, her daughter is Olympic champion. And nobody said that Kati who works with mRNA. Thank you.
So I would like to continue. It's likewise a extraordinary honor here to be awarded with the colleagues and the young scientists. About 17 years ago, we had the pleasure to be a young scientist honored by the German Immunology Association. We got the George Köhler Prize in 2005, and I know how you are all feeling today. It was such incredible bonding to the society, and this bonding never ended. So I would like to tell the story from our side. We were on the other side of the Atlantic, and we started to work in 1996 on mRNA, and Kati has mentioned that hundreds of scientists has contributed to the understanding of mRNA and the elements, and among the scientists who inspired us was the work of Eli Gilboa. Yeah, we were at that time point in the mid of 90s. We had discovered tumor antigens with a new approach, and we were learning that these tumor antigens are recognized by antibodies and by T cells. But we knew that these immune responses were extremely weak. And one of the non-so-motivating discoveries at that time point was that every patient had other type of tumor antigens, and we were interested in developing an approach for cancer vaccination to induce T-cell responses. We started with DNA that was at that time on vogue, with implying strong immune responses in mice, but we stopped our work after 1 year, after we understood that DNA plasmids don't work in human dendritic cells. And we went on with mRNA. At that time point, Eli Gilboa's group had described an approach taking mRNA, transfecting that in vitro into dendritic cells, and re-infusing this dendritic cells to animals. A few years later, this was described also in the human setting. The approach in principle induced immune responses, but the immune responses, the quantity of the immune responses, were about 100 to 1,000-fold too low to fight cancers. So we were interested to overcome this by using mRNA for direct injection in vivo, and ideally reaching immune cells in vivo, and came up with a strategy to systematically identify how to increase the stability of the mRNA, because when we injected mRNA, the amounts that were needed were extremely high to induce an immune response. And so we systematically analyzed the poly-A tail, the cap analogs, 5' UTR, 3' UTR. This started in the end of the 1990s, beginning 2000, later on in Christoph Huber's department, and we found rules how to systematically improve the mRNA. What we found out is that if you integrate independent improvements, with each improvement coming up with a increased translation of factor 3 to 5 times, and if you then combine them, you have an amplified improvement, resulting in increased stability, increased translation, and increased durability of expression. So another finding that we made, which was instrumental, is that taking the mRNA and injecting to different routes, we identified that injection into the skin, injection into the muscle, injection into the dermis resulted in some immune responses. But when the mRNA was injected in the lymph nodes, we saw dramatic increase of immune responses, factor 100 higher than in the other routes. We investigated that deeper and found that the mRNA in dendritic cells are taken up in the lymph nodes are taken up by dendritic cells. Even after 5 minutes, you could see an uptake by the dendritic cells, and after 30 minutes, the mRNA ended up in the dendritic cells. And this mechanism, which is driving that, was micropinocytosis. And we asked the question: can we deliver mRNA in a way that dendritic cells could take that up? This is our experiment which we had performed in 2004, published in 2010, showing just the increase, the dramatic increase of the immune response after injecting the mRNA into lymph nodes with T cells here stained with Taiwan 2, yeah, and was injected with control mRNA, and this is mRNA encoding for the right antigens and showing the expansion of CD4 and CD8 T cells. So we started to systematically analyze how to develop lipid formulations that allow us to deliver the mRNA into dendritic cells in lymphoid tissues, and discovered that certain physical chemical properties of lipid nanoparticles, that means lipid nanoparticles of a slightly negative charge, deliver the mRNA directly to dendritic cells in spleen and in the lymph nodes in the spleen to the marginal region. And this is based on the uridine mRNA. We explicitly focused on immune stimulatory TLR7, TLR8 activating mRNA. This dendritic cells, after uptake of the uridine mRNA, were stimulated and migrated to the T cell zone. And this recognition of the TLRs resulted in an interferon alpha response, and this is what we need to get really, really strong T cell responses of Th1 type with maturation of dendritic cells, resulting in dramatic increase of immune responses. So we translated this approach into the clinical setting and use various mRNAs that have been tested in the past, for example, New York ESO and tyrosinase, to evaluate whether with this type of mRNA we get much stronger immune responses. We could show that this lipid nanoparticles in humans behaved in the same way as in mice. They were targeting the spleen and they were inducing type 1 interferon response. This is from the first clinical trial. It was the first lipid nanoparticle clinical trial worldwide. The regulators asked us to start with doses at the level lower than we had used in mice. This is just 7 microgram of mRNA injected into patients, and then with weekly dose escalation, we reached doses of 75 microgram, showing a linear increase of type 1 interferon. And what was the most amazing observation was that for the first time we could see T cell responses in the range of several percent, in some patients up to 20% of peripheral CD8 T cells were antigen specific, and this was associated with objective responses in a proportion of patients with vaccine alone and in combination. So we translated this approach into our original approach, because as I started, the challenge for cancer immunotherapy is that every patient has a different tumor, and even within the tumors there is a heterogeneity. The question is, how can we address this heterogeneity? And the basic idea was to target mutations. So we started to develop an approach which is based on following steps: get the tumor sample from the patient and blood sample, perform a sequencing, next generation sequencing. So in 2010, we got the first sequencing machine in Germany. The other 80 machines went to China. So this machine, before we could start this machine, we had a problem because our labs were below the helicopter landing place. And every time when the helicopter landed, we had a difficulty to get the sequences, because then there was the noise. So we had to find a way to put the machine on a non-vibrating table. So with the first sequences, we started this project in 2010, and in 2014 we were able to go into the clinic. And the project is based on the discovery of mutations by comparing the exome DNA from blood and tumors, identifying the mutations, and using the messenger RNA sequencing to evaluate whether the mutation is expressed. We use prediction algorithms to ensure that we can select the mutations that are recognized by CD4 and CD8 T cells. This was started by simple algorithm is now in the phase where we use machine learning and AI algorithms. And then we created our vaccines by just selecting the mutations and bringing that back to our uridine optimized mRNA backbone, and then using this mRNA to treat patients. So in our first clinical trial, which was published in 2017, we could show that every patient developed immune responses against multiple mutations. We could show that mutation-specific immune responses could reach levels up to 10% of peripheral T cells, and we could demonstrate that these T cells enter tumors and are able to kill cancer cells. We saw that the vaccinated patients had a dramatic decline in their metastatic relapse rate after vaccination, and based on this, we started to evaluate how to bring this type of treatment into a broader setting. The first clinical trial was in melanoma patients. We know that melanoma is an excellent tumor for cancer immunotherapy, but the question is whether this personalized vaccine could also work in other tumor indications with lower rate of mutations, for example, in pancreatic cancer or triple-negative breast cancer. In the meantime, we have shown that this is really working independent of the tumor type. This is our study, the study in triple-negative breast cancer patients. We see again that we can induce in all patients immune responses with an extremely high magnitude. These T-cell responses are stable for up to 2 years and are associated with control of metastatic relapses. We have similar data in pancreatic cancer, where we see that in this case only 50% of patients responded with a vaccine response, but this 50% of patients do not have a relapse after surgery, whereas those patients who did not respond to vaccination had a relapse, indicating that immune responses are associated with vaccination. Our goal is to bring that into early stage of tumors. We don't believe that these vaccines will work in advanced cancers, but the medical need is after surgery. We know that, for example, in pancreatic cancer, about 70% of patients relapse after a few years. In triple-negative breast cancer, it's about 40%, and even in colorectal cancer, it's about 20 to 35%, depending on the clinical stage. And the idea is, after surgery, get the tumor sample, do the sequencing, and provide a personalized vaccine for these patients, and thereby decrease the metastatic relapse rate. For this, we have invested a lot into process optimization. When we started this was a huge effort. So this is the paperwork before we got regulatory inspection in Mainz. So data from a few patients. It was all paperwork. Each manufacturing for every single patient was about 1,400 pages of documentation. Every manufacturing was done in a manual version, and of course this is not viable. We went and automatized that. We have now a digitalized approach that allows us to develop this, to produce these vaccines in less than 6 weeks. And we had done that for hundreds of patients until in 2020 the pandemic came.
So fast forward to that fateful weekend in January 2020, when we realized that we were actually already in the midst of a pandemic, and we were convinced that the world as we knew it would change. And you have heard about the science, the technology reviewed by Uğur and Kati. We believed that what we had worked on in all those years and also clinically translated had the potential to allow us to contribute to the response of the pandemic. And therefore we decided to kick off Project Lightspeed directly in order to help to contribute to a vaccine development. Already at that weekend, 20 different candidates, vaccine candidates, based on different versions of our mRNA toolbox and different variations of the spike protein were generated. These were funneled through the usual lab experiments in a comparative manner. 3 months later, we had selected the 4 best performers, which were introduced into our phase 1 trial. Again, 3 months later, we have selected, based on the clinical data, safety and immunogenicity, the candidate which was the most promising and encouraging one, BNT162b2, aka Comirnaty, and started a worldwide phase 3 trial in 45,000 participants. And in November 2020, 10 months after starting making the decision to start this program, we were able to report that our vaccine had a vaccine efficacy of 95% against symptomatic COVID-19 disease. We had planned for success at risk, because this is what you do in a desperate situation, meaning that we had forged alliances with partners with complementary skills and capabilities, with Pfizer, for example, who became a key partner. We had initiated conversations very early on with all regulatory authorities around the world, and at risk submitted already our data. And this, and therefore we were able to get the first approvals within that year. In the meantime, Comirnaty has been administered to more than 1 billion people in more than 170 regions, and we hear that millions of deaths were averted. BNT162b2 is the candidate which we selected as a single-stranded mRNA. It encodes for a conformationally stabilized version of the full-length spike protein of the Wuhan Hu1 wild-type virus. We have introduced all those non-coding design elements which we have developed over the years, which Ugo has featured. The vaccine is, the mRNA is enveloped in a different lipid formulation as compared to the one we use in cancer, which is intravenously administered. Pandemic vaccines are administered intramuscularly. This formulation, lipid formulation, which is different to our cancer vaccine, comes with its own intrinsic adjuvanticity and the chemistry of our mRNA to balance the innate and the adaptive immune responses is based on nucleoside modified mRNA. So everything which has been developed in all those years is combined here. The design principles for the RNA lipid nanoparticles are based also on our research. So we used design principles which would allow us to bring the lipid nanoparticles and the RNA to the place which is of interest, resident dendritic cells resident in lymph nodes. And you can, is this a pointer? No. And you can, sorry, how can I go back? And you can see this here on this slide, the top panel, RNA after injection into the muscle, and on the bottom, the protein, the IHC staining of the protein. You don't see signals in the muscle, but the nanoparticles migrate to the draining lymph nodes and beyond. The mRNA is accumulated in the lymph nodes, and the protein is expressed there. In mouse experiments, we selected this candidate because we saw that an immune effector response, both B cell and T cell, is initiated. We get binding antibodies which are neutralizing and high affinity, a CD8 T cell response and a Th1 type CD4 response. We translated then this into our phase 1 trial, and in our phase 1 trial we observed that already low dose levels between 10 and 30 microgram administered 2 times 21 days apart lead to neutralizing antibodies, which neutralizing antibody titers which are comparable to a neutralization of convalescent sera, and also to T cell responses, CD4 T cell responses, as well as CD8 T cell responses, which are comparable in magnitude with what we see for recall responses against frequent viruses. And this all translated into vaccine efficacy. The early efficacy data was, as I said, 95% across ages, including the older adults, and it was persistent. After 6 months, we still had high efficacy, in particular the efficacy against severe COVID-19 disease, which we think depends on the strong CD8 T cell response we could induce, remained pretty high. The challenges were not only scientific. One of those non-scientific challenges was supply. Ensuring that we could manufacture the amounts we needed for the huge clinical trial program, but later on also for supply for the pandemic rollouts. In 2019, having a scale which was meant for cancer vaccines meant 10,000 doses per year. In 2020, we managed to scale up to 25 million doses, realizing that a pandemic vaccine against a virus needs a different scale. And thereafter, realizing that many of those peers who started with us with the intention to also chime in into the vaccine development did fail or could not continue. We understood that we also had the obligation to ensure a broader supply, and were able to even upscale more at the end of the day, a scale up factor of 300,000. The mission did not end with having a vaccine to supply. It was very clear from the very beginning that the virus would mutate, and we wanted also to contribute to setting up continuous preparedness and a response to the continuous, fastly evolving virus. And for this purpose, we developed a machine learning-based early computational detection system to assess virus variants with regard to their fitness, but also to their risk of immune escape. And what we did with all those variants of concern which were identified was to test them with sera of BNT162b2 immunized individuals for proficient cross-neutralization. For the first part of the Greek alphabet, so with your alphas and deltas and lambdas, we did not worry because we saw cross-neutralization. However, when Omicron came up, we could see in the neutralization assays, as you can see up there, that neutralization was not robust, was not any more robust after the primary 2-dose series, but we had to boost to ensure still high neutralization levels. And so that means you can continue to boost to cover an immune escape variant, but at some point it makes sense to develop a variant-adapted vaccine. And this was in the basically next milestone into which we embarked in November last year. And this was not really a challenge for us, because you have heard from Uğur that we had run this race of adapting to a variant, namely to a cancer variant, hundreds of times with our personalized cancer vaccines, and we just needed to copy this process for variant adaptation of the vaccine. There, however, was a challenge, as it turned out, namely that we need to work with all the stakeholders on finding proficient regulatory pathways for such variant-adapted vaccines. The interaction with the regulators led to approval process with clinical testing, which took 8 months, also at light speed 8 months. This was not feasible faster. And you all have experienced that the sublineages of Omicron evolve very fast, so that basically every 3 months you have a different variant. And therefore, these timelines mean that at the point where you have a variant-adapted vaccine approved, you already have not the next, but the 2 next variants on your table. And therefore we proposed, and regulators listen, an approach which is close to what we do with our personalized cancer vaccine, a path which only takes 3 months. But there were more areas where we found we have the obligation to contribute. As a small company developing cancer vaccines, we had not much exposure to inequality of distribution of vaccines or innovative medicines. And having developed a COVID-19 vaccine, we clearly saw that there is this inequality. There are different ways of reacting to that. One immediate solution which we could provide was that we delivered our vaccine at cost to low and middle income countries, and 40% of our vaccine deliveries went there. But this is not really sustainable. You would want those regions, those people to have the opportunity to produce for themselves, meaning technology transfer, which is not an act but an attitude. And for this we developed, can I activate this movie? Yes. And for this we developed the biocontainer concept. So a miniaturized production plant which is assembled, and test runs are performed in our facilities in Germany. And these containers include everything: automatized production, air purification systems, cloud-based manufacturing in one container. After assembling, after test runs, after installing everything, they can be transported to any place and set up there and certified directly, and can then produce at those locations. We just had the groundbreaking event in Kigali, Rwanda, which is the first place which will have these containers, and we hope with this also to ensure that in those regions people can get access to the COVID-19 vaccine and its adaptations, but also other vaccines we plan to develop. With this, I would like to thank the funding agencies who have supported us, in particular also in our academic settings. Those people, and this is only a selection of all those who have been involved in our cancer vaccines and those who have been involved in our COVID-19 vaccine development. And I would like to thank you for your interest and reiterate how honored we are to receive this prestigious award.
There's nothing more to say. Thank you so much for this insight of all the developments. I can mention at this point that this session, as well as the others, are recorded. And you will be able to watch that in our Mediathek. And maybe you just go home to your institution, take your students who are not here, and just watch the 3 presentations, because I think it's worth getting the entire story, and it's very rarely that there's enough time to really go through the entire development, and this is a piece where we are very happy that we will have that in our Mediathek. Thank you so much. You will have to leave now. I promised that I can leave you out here at 7:30. We are in time. I really promised that. So please excuse that they have to leave now, but we're very happy and honored that you have been here. Thank you very much. So, we're almost done. You may have realized that when we started as 5:15, I had 2 messages where my blood pressure went up sky high and it never came down to baseline. The first message was, we do have a surprise awardee at the end of the session, and the message was he was not going to make it, but he's here. He has maybe an idea that something is going to happen tonight, but he had no idea what this was. I think you may not know him personally, but you certainly know the company. It's BioLegend. So we decided already 2 years ago that Jean Lay, the founder and CEO of BioLegend, will receive the honorary medal of the German Society of Immunology. And do you know why? Of course, it's a company, and I think all of us had spent a fortune on BioLegend. That's not the point. What makes the difference is that, Jean, when we were in Munich 2019, it was crystal clear that you do support not only the German Society of Immunology with our meetings many years back, you also do that with the Italian Society of Immunology, the French Society of Immunology, the Swiss Society of Immunology, and many others. The difference is that without you, we would not have been able to do a lot of things during the meetings in the last 15 years. And we thought that this is something where we give you something back to your continuous support of our society, but also the others. So Jean Lay, we are very happy that you made it, and you will receive the honorary medal from Carsten Watzel and myself for your continuous support of our societies. Thank you very much for coming.
Few words. Yes, I hold this for you. Thank you, Christine. I'm very, very honored to receive this award. This to my surprise, because I did not know anything. But all I know is I want to be there, because this is very special. I want to give my tribute to Dr. Reinhold Schmidt, because I met him 2009. I came here, Hannover, to meet him, because he is the ECI, second ECI meeting president. Because I want to express to him, I want to be the sponsor to that meeting. And I and Reinhold, and also Roland Jacobs, if you are here, if you remember, and we were in a restaurant just not too far away from here, the school here. And when we talk about the BioLegend mission, we want to enable scientific discovery to therapy. And that's we have a very common, because he is in the clinical immunology. So we have very common mission. So then, since 2009, sponsor ECI meeting, and then a DGfI meeting every year till now. We'll continue on that. So I want to pay my tribute to, to Dr. Schmidt. And secondary, I just thought, I made it because, you know what, my flight was delayed and then missed the connection. So my colleague quickly take a train from Frankfurt to here. We missed the first part of that, but at least we are able to join after 6. And I saw the young investigator for the George Kohler Prize. I want to share with you, he is my inspirational person, not only him as well as Caesar Milstein. Why all my career from then? Because they published a paper in 1975 regarding the hybridoma technology. I can make a large scale of monoclonal antibody potential if carry the drug specific to cancer and then cure cancer, and then cure cancer disease. I said, wow, I was inspired by that because I was in my senior year in veterinary medicine and supposed to be the animal doctor, but I changed my career track to biotechnology. All my career for 40-some years, all in the monoclonal antibody development. That's why I become the BioLegend, all the contents. So I want to share with everyone here, that's how my story is a good story. How can we translate technology, scientific discovery, to therapy or to diagnostic, or even advance more new scientific discoveries? Catalin's story, I'm inspired by her story very, very much. I thought that she's going to stay here. Really, really, I can pass my baton to her because she's much more successful than BioLegend. I guess that's I'm not a good speaker, but I want to show from my heart. Thank you. Thank you for the award.
So we are almost done. The second message we got 5 o'clock was that our caterer made a mistake and there would be no single piece of food and no single glass of wine or water outside when we are done at 7:30. In the background, the team of KIT with Janet Jambor and my colleague Sonja Klein made the impossible thing possible. There will be food outside for the welcome reception. I have no idea what will be outside. They just texted me and said, we got food for you, we got drinks for you in the building J1 and also in the building J2, where there are the posters and the other lecture halls. So if it doesn't taste, blame it just on me, because it was just a sort of instant action within 2.5 hours to get something to eat and drink for you. We will definitely do that differently on Friday night at the Immunonight, I promise. So whatever is outside, just take it. Enjoy this evening. Enjoy that we are back together, and all the rest, I think, is secondary. And I thank you very much for staying so long. I think it was a terrific afternoon. Thank you so much.